Okay, everybody, welcome to the back end of Chapter 3, and we're going to talk a little bit more about membrane transport, and here we're looking at selective permeability, and this is the ability of the plasma membrane to allow certain materials to pass in and out of the cell membrane while excluding others, and this influences movement both into and out of the cell. Essentially, a cell doesn't want to let just anything in or out in an effort to um, control activities related to metabolism, which consist of the building of macromolecules using an energy input such as ATP, or the disassembly of molecules in order to extract energy in order to carry out vital cell functions. If we just let anything into the cell and anything out, what would happen is that we wouldn't have conditions that would promote metabolism. So there are several different mechanisms that the cell will use in order to transport stuff in and out of the cell. Membrane transport is defined as movement of substances into and out of the cell. The cell membranes are selectively permeable, meaning that some substances can pass through while others can't. And there's two basic modes of transport. One is passive transport, where no energy is required in order to move the substance across the cell membrane. And the other is active transport, where the cell has to provide energy so that it can move the substance in any direction it wishes, even if it is not energetically favorable. It doesn't move along with its concentration gradient. And you'll see what I mean as we move on to our first process here. This is diffusion. And diffusion is something I'm sure you're all familiar with if you've ever seen a smoke ring diffuse as for somebody blows it from smoking a cigar, and it eventually fills the entire room. This is the property where particles tend to distribute themselves evenly within a solution. The movement is always from a high to low concentration, or what we call down a concentration gradient. Uh, this doesn't require any input of energy from any other source. What drives this movement is simply the difference in concentration between the source and where the source is going to eventually move to. So you can see an example of diffusion here as this tablet begins to slowly dissolve inside the water and eventually disperse itself evenly to all parts of the solution. You don't need to put any energy into this in order for it to happen. So these are passive processes. Simple diffusion is an unassisted process. The solutes are lipid soluble in order to pass through the cell membrane, which, remember, is made up of both phospholipids and also interspersed with cholesterol and proteins at certain strategic points. But these molecules can pass very easily across the cell membrane because of their small size, and also the fact that the lipid barrier, the central portion of the cell membrane, doesn't provide much of an obstacle to these molecules getting either into or out of the cell. So they simply flow down their concentration gradients from where they're most concentrated to where they're least concentrated with no other energetic assistance. Examples of molecules that can do this include water, oxygen, hydrogen, and lipid-soluble molecules, things like cholesterol and catecholamines such as epinephrine can pass very easily through the cell membrane down their concentration gradients without any added assistance. Osmosis is simply referring to the diffusion of water down its concentration gradient from where it's most concentrated to where it's least concentrated. The polar water molecules easily cross the plasma membrane through aquaporins, but they're also capable of moving directly through the cell membrane across the lipid bilayer. So what this does is this dictates how much water a cell can hold. The reason this is important because water provides an environment in which chemical reactions can happen. So the cell has to control how much water it contains in order for it to maintain the processes that keep it alive. Okay, so very important that we have osmosis as an operating principle. And again, this is simply concentration gradient driven. Okay, the water, for instance, in this illustration is highly concentrated outside the cell and not so much inside the cell, and as a result will flow through the cell membrane. Facilitated diffusion is a little bit different. In facilitated diffusion, the molecule is too large or perhaps too polar or charged in nature to pass through the cell membrane directly, and so it needs assistance in the form of a protein carrier. So these are substances that require a protein carrier for passive transport. And examples of this type of transport include lipid-insoluble substances and large substances. And so you can see some examples of carrier-mediated facilitated diffusion through a protein carrier. Here's glucose, for instance, being carried across the cell from where it's most concentrated to where it's least concentrated. And here you can see an example of perhaps a charged particle passing through a channel from the outside to the inside of the membrane down its concentration gradient. So again, no energy is required for this to happen. It happens automatically. Filtration is a little bit different. In filtration, water and solutes are forced through a membrane by fluid or hydrostatic pressure. In order for the solutes to move across the membrane, a pressure gradient has to exist, meaning there has to be an area of high pressure on one side of the membrane and an area of low pressure on the other side. The solute-containing fluid is pushed from a high pressure to a low pressure area. An example of where this happens in the human body is in the kidneys, where blood that's pumped through the body by the beating action of the heart is cleaned in special structures in the kidneys called the nephrons. And particularly, there's a structure inside the nephron called the glomerulus, which contains blood vessels that have very tiny slits in them, just big enough to let molecules below a certain molecular weight pass through. And what drives them through those holes is the pressure gradient more so than the concentration gradient, even though both processes are active at the same time. An example of this would be, say, filtering your coffee after you've put it in the coffee filter, okay, where the water and the coffee flavor comes through the filter and the grounds stay held up by the filter. And the only reason that the coffee drains through the filter is because of the force of gravity, which generates a hydrostatic pressure that causes the coffee to go into your cup. Okay, so that's one example of filtration. Now, active processes are different. In an active process, we can move a molecule in any direction that we want because we have energy to drive the process. So these are substances that are transported unable to pass by diffusion. The substances may be too large. They may not be able to dissolve in the fat core of the cell membrane where the lipid component of the phospholipid is housed. Or substances may have to move against the concentration gradient. All of these types of tasks can be helped using ATP, which is the universal energy currency, to drive what would normally be an unfavorable process. So there's two common forms of active transport. One is active transport known as solute pumping. The other is vesicular transport, and under vesicular transport we'll examine exo and endocytosis, as well as phagocytosis and pinocytosis. So the active process, active transport or solute pumping, is responsible for moving amino acids, sugars, and ions via protein carriers known as solute pumps. The ATP energizes the protein carriers, and in most cases the substances are moved against concentration gradients. And you can see an example here of an active pump at work. This is an example of a very important pump in living systems, particularly in the human body, called the sodium-potassium ATPase. And what it does is it binds the energy molecule ATP and uses the energy from that molecule to force 3 sodium ions out of the cell for every 2 potassium ions that it brings back in. And again, these both happen against their concentration gradients. Okay, so what we're doing here is we're moving substances in an unfavorable direction. What you have to realize about this is that the reason this can occur is because of ATP as an energy source, and the importance of this particular pump is that it serves to allow the membrane to act as a sort of battery for the cell, a type of stored energy in which positive and negative charges are separated from each other, where the inside of the cell becomes negative compared to the outside, due primarily to this pumping action that's driven by the hydrolysis of ATP, the release of energy from ATP. And the cell can harness this energy to do all kinds of other things. It can harness that energy to transport stuff. It can harness that energy to move things in and out of the cell, either with or against their concentration gradients. This is a phenomenon known as membrane potential. We see this at its most important in cells that have a property known as excitability. Examples of cells that are excitable include neurons and muscle cells. Both of these respond to an electric stimulus by producing an electric current that's moved primarily down the cell by the movement of these ions across the cell membrane. Okay, so without this pump working, we wouldn't be able to have a functioning nervous system or a functioning musculoskeletal system. So this is a very significant biological entity. The sodium potassium ATPase is found in every cell in the body. Okay, another type of active process is vesicular transport. What a vesicle is basically a membrane bubble that closes up around something. Could be a solid particle, could be some sort of cellular product, could be something that we want to bring into the cell to use perhaps as fuel. There's a wide variety of potential solutes that can be transported using this type of method. It requires energy. In exocytosis, what happens is that materials move out of the cell from inside the cell. The material is carried to a membrane vesicle, which migrates to the plasma membrane. The vesicle then combines with the plasma membrane, and the material is emptied outside the cell and then is used to do something. Okay, could be a signal molecule, could be some sort of extracellular matrix protein that helps hold cells together, okay, such as you see in connective tissue. A wide variety of possibilities for vesicular transport, exocytosis, exist. What you're looking at here is just one example of vesicular transport using the snare T-snare mechanism. And what happens here is that the membrane-bound vesicle migrates to the plasma membrane. Then the V and T snares bind to each other. They recognize each other. The plasma membrane is then helped to fuse with the vesicular membrane, and the result is that the contents of the vesicle are released into the exterior. And then by the same token, this process can reverse itself, and the vesicle can recycle to go pick up more material and bring it back outside the cell. Okay, so this is a process that's again driven by ATP. It's not something that would happen automatically in the cell, but it's very useful because it aids in an abundant number of life processes in some of the different systems that we'll examine. Cellular secretions are very important to their function. Here you can see another type of vesicular transport. This is endocytosis, where extracellular substances are engulfed by being enclosed in a membranous vesicle. Different types of endocytosis include phagocytosis, which we call cellular eating, where a solid particle is brought inside the cell, enclosed in a vesicle, and then either transported through the cell or perhaps fused with a lysosome in order to break down the components of the solid particle and perhaps use them for energy or other purposes. And in penocytosis, instead of a solid component being brought inside the vesicle, we bring liquid inside and transport it either through the cell or we transport it to some location inside the cell. And so you can see an example of penocytosis here, right? The lysosome, the vesicle fusing with the lysosome for digestion of the ingested substance. The detached vesicle contains the ingested material. Then the cytosolic vesicle fuses with the lysosome, and then we recycle this vesicle back to the cell surface in one pathway, or we can actually dump the contents on the other side of the cell, depending on the needs the cell has for that particular substance. Here you can see an example of phagocytosis here, the solid particle being engulfed, and here is penocytosis, taking in a substance from outside the cell to transport it either across the cell or to some location inside the cytoplasm. Now the cell has a life cycle as well. At some point the cell is going to have to go from being one cell to being two once it reaches a certain stage in its growth. The cell carrying out its normal role in the human body is said to be an interphase, and during this period the cell will grow and carry out metabolic activity. It'll derive energy from stuff that it ingests, and it'll use that energy to build and repair things and also to grow. In cell division, the cell is going to reproduce itself, and the function is to produce more cells for the growth and repair process. So two general events happen in the cell life cycle. One is mitosis, which is the division of duplicated genetic material, and the other is cytokinesis, which is the division of the cell itself from one cell into two. And so what you're looking at here is one of the major components inside the cell. This is DNA, and the significance of DNA is that it's an information molecule. And what I mean by that is that the building blocks of DNA, which are called nucleotides, can be arranged in any length and in any order. And because of that, the amount of information they can carry is almost infinite. A particular stretch of DNA that contains the instructions for building a particular type of protein is called a gene. And there are thousands of genes inside every human cell that contain instructions for protein products that help that cell carry out its role, its normal role in the human body. When and where those genes are used depends on the cell's needs for those products at any particular time. The genetic material, as a result, is vital for the cell's survival, and when the cell gets ready to divide, the first thing that happens is that this double-stranded DNA molecule will unwind, and when it unwinds, it'll reveal the information that's contained on both strands, and enzymes, which are proteins that in this case help build DNA, will come in and assemble a new DNA strand using the instructions contained on the old DNA strand. One of the things that you'll notice, for instance, in this figure is that for every nucleotide labeled C, on the opposite strand there is a G. For every nucleotide labeled T, on the opposite strand there's an A. For every A there's a T. For every G there's a C. And so the information contained on one strand of DNA is reflected by what we call its complement on the other strand of DNA. And that's why DNA has this unique ability to reproduce information and carry it from one cell to the next to the next. So as this DNA molecule unwinds inside the nucleus of the cell, enzymes come along and use this information on one of the strands to build a new strand of DNA, and by the time we get to the end of the DNA molecule, what we have are two brand new double helical strands of DNA that each contain the exact same information. What will happen next is that that genetic information will bundle up into visible structures called chromosomes that you can see under a microscope if you stain the cell's nucleus. And those duplicated chromosomes are then going to be split in half in the process of mitosis, and each new cell will get one copy each of each individual chromosome. This replication of DNA generally occurs towards the end of interphase, where the DNA uncoils and each side serves as a template for the new strand. So what we're going to watch next is the process of mitosis. There's a couple of videos I think that'll be very useful in explaining what happens when a cell gets ready to divide. So stay tuned and listen to these mitosis spots.
Cell division consists of two processes, mitosis and cytokinesis. Mitosis, division of the nucleus and its chromosomes, is divided into 5 phases: prophase, prometaphase, metaphase, anaphase, and telophase. Mitosis is followed by cytokinesis, when the cytoplasm splits to form two separate daughter cells. The cytoplasm of a cell in late interphase contains two centrosomes, each of which may contain a pair of centrioles. In the nucleus, the chromosomes have been replicated during S phase, but are still dispersed in the form of chromatin. One or more nucleoli are present. During prophase, the nucleoli disappear and chromatin fibers coil up to become discrete chromosomes. Each chromosome consists of two identical sister chromatids, joined at the centromere. Microtubules grow out from the centrosomes, initiating formation of the mitotic spindle. During prometaphase, the nuclear envelope breaks into fragments. Some of the spindle fibers reach the chromosomes and attach to kinetochores, structures made of proteins and specific sections of DNA at the centromeres. Non-kinetochore microtubules overlap with those coming from the opposite pole. At metaphase, the mitotic spindle is fully formed, and the microtubules attached to kinetochores move the chromosomes to the metaphase plate, an imaginary plane equidistant from the poles. Anaphase begins when the two centromeres of each chromosome come apart, separating the sister chromatids. Once separate, each sister chromatid is considered a full-fledged daughter chromosome. Motor proteins of the kinetochores walk the daughter chromosomes along the spindle microtubules toward opposite poles, and the microtubules shorten. At the same time, the spindle microtubules not attached to chromosomes lengthen, pushing the two poles farther apart and elongating the cell. During telophase, nuclear envelopes form around the identical sets of chromosomes at the two poles of the cell. The chromosomes uncoil and nucleoli appear in the two new nuclei. Meanwhile, cytokinesis begins, splitting the cytoplasm and separating the two daughter cells. In animal cells, cytokinesis begins with the formation of a cleavage furrow. At the site of the furrow, a ring of microfilaments contracts, much like the pulling of drawstrings. The cell is pinched in two, creating two identical daughter cells. In plant cells, cytokinesis begins when vesicles containing cell wall material collect in the middle of the cell. The vesicles fuse, forming a large sac called the cell plate. The cell plate grows outward until its membrane fuses with the plasma membrane, separating the two daughter cells. The cell plate's contents join the parental cell wall. The result is two daughter cells, each bounded by its own continuous plasma membrane and cell wall.
This soccer player's knee will be as good as new in a few weeks, thanks to mitosis, a type of cell division that generates new cells for growth and repair. Let's move into a cell to witness the events of mitosis. Before a cell can divide, it must first duplicate the chromosomes stored in its nucleus. During chromosome duplication, several bubbles open up along the chromosome. Each bubble grows until it merges with an adjacent bubble. Each chromosome now consists of two identical copies called sister chromatids. Getting closer, we see that each sister chromatid consists of DNA wound around small proteins called histones. The sister chromatids begin to coil into tight helical fibers. Outside the nucleus, centrosomes that duplicated earlier move away from each other to opposite sides of the cell. Microtubules extend from the centrosomes, forming the mitotic spindle. Back in the nucleus, the DNA forms loops, becoming more compacted. These structures fold back on themselves, eventually condensing into a shorter and thicker chromosome consisting of two sister chromatids. As the chromosomes continue to condense, the nuclear envelope breaks up. The array of spindle microtubules is now extensive, and the chromosomes are fully condensed. Spindle fibers from each pole attach to protein structures located at the centromere of each sister chromatid. As the chromosomes are bound by spindle fibers from opposite poles, they move first one way and then another. The counteracting forces of the spindle eventually cause all the chromosomes to end up at the center of the cell, as if arranged on an imaginary plate. The sister chromatids are released from each other, each becoming a full-fledged chromosome. They are moved toward opposite poles of the cell, pulled along the spindle fibers attached to them. At the same time, overlapping spindle fibers that are not attached to chromosomes continue to lengthen, pushing the poles farther apart. Once the chromosomes arrive at their destination, they become less condensed. Two new nuclear envelopes form, completing mitosis, the division of one nucleus into two genetically identical daughter nuclei. The cytoplasm divides by the process of cytokinesis, forming two separate daughter cells. In your body, millions of cells divide every second, providing new cells for growth and for repair of damaged cells.
Okay, now that we've seen how it is that cells reproduce, first by copying their genetic information and then by evenly apportioning that genetic information to the new cells, we're going to move up now to the next level of organization that we dealt with in lab last time we were together, and those are tissues. And the way to look at this is that we're moving up a hierarchy. Okay, we've discussed to some extent the particles that make up all the matter in the universe. Those are the atoms, and we've also touched on the fact that when atoms combine with each other, they produce molecules. Molecules can combine with each other to make macromolecules, and some of the most important macromolecules in living systems are polymers. Some of those important polymers include proteins, DNA, and RNA. Okay, the proteins being the workhorse of the cell, and the DNA and the RNA being information molecules. And these macromolecules can then be assembled together into structures called organelles, which can exist inside cells, which are the smallest units of living matter. And those cells have the ability at certain points in their growth cycle to reproduce themselves by first duplicating their genetic material and then dividing in half. And in the early part of the Earth's history, that was what most life forms were single-celled organisms. And life was relatively uncomplicated at that point because in order to get food, all the single-celled organism had to do was transport it across its cell membrane, and in order to generate waste, all the cell would have to do is extrude that waste into the extracellular environment using processes like exocytosis and passive transport. But something happened along the way, and as the Earth began to change, as parts of the Earth began to get cold, parts began to get warm, parts began to get dry, parts began to change their chemical composition, life had to either adapt or die. And so one of the first adaptations we think went on in living systems was the change from single-celled organisms to multicellular organisms. And the thinking is that in Earth's early history, a lot of these single-celled organisms that were particularly good at performing certain functions would gather together and would serve as a community of single-celled organisms that would perform a particular task for a larger community of single-celled organisms. You could imagine, for instance, that one group of single-celled organisms might be very good at fighting off attacks from outside invaders. Another group of organisms might be very good at gathering food from the external environment. And another group of organisms might be very good at building a shelter, so to speak, for the rest of the community to live in. And these communities are still in existence today. They're called biofilms, and they're made up primarily of bacterial cells. But in the Earth's early history, we think that the cells that would eventually become the organizing units of organisms that are a lot like human beings, eukaryotes, also had this sort of loose association. And the next leap then was the idea that these loose associations of cells became so dependent on each other that they couldn't live without each other, and they formed the first tissues, which are groups of cells with a similar structure and function. There's four primary types of tissues that we see in living systems. The types that we're going to touch on in our study of the human body include epithelial tissue, connective tissue, muscle tissue, and nervous tissue. They all look very different. They all have very different roles, but they all have exactly the same genetic material inside the nucleus of each cell type. And so you might well ask, well, how is it that with the same genetic material, each one of these cells can look and act so different from each other? You may have noticed, for instance, in lab, when you were looking at muscle tissue and nervous tissue and connective tissue, that they were all very different from each other in appearance and in location and in the role they had in the human body. And the explanation for that is that the genetic information contains all the instructions necessary for every cell in the human body to perform its task. But as the human being develops from a single cell to a multicellular machine, what happens is that certain cells are restricted to only certain sets of instructions inside the DNA. In other words, they're not allowed to read from certain parts of the instruction manual, and what this does is this shapes how they look, how they act, where they're located, and what their role is. So for instance, as the human being develops inside its mother's womb, the epithelial cells will eventually only be allowed to read from the epithelial cell handbook of DNA, the connective tissue only from the connective tissue handbook in the DNA, the muscle tissue and the nervous tissue reading from their respective locations in the DNA. And those locations are scattered throughout all the chromosomes, okay? But because of this selective use of information that's in the nucleus, we have a profound impact on how these cells behave, what they do, and what they look like. So let's start with epithelial tissue. Where do we find it? We find it in body coverings and body linings. We also find it in glandular tissue. Glandular tissue is tissue that's tasked primarily with producing secretions that the body uses in order to maintain homeostasis. And its functions include protection, absorption, filtration, and secretion. Okay, so when we say protection, we mean guarding what's inside you from what's outside you, or guarding what's inside you from getting outside of you. Okay, an example. Okay, your epithelial tissue on the surface of your skin protects you from pathogens that might want to get deep inside your body through ports of entry, okay, say punctures or lacerations, openings in the skin, and so forth. If we didn't have a nice layer of stratified squamous epithelial tissue, okay, we would be prone to a lot of infections and disease, and it'd be very difficult for us to maintain homeostasis. Another example of protection, the stomach is an organ in the digestive system which digests food by producing secretions that are very corrosive. And if we didn't have the protective epithelial lining of the stomach, then those secretions could get outside the organ and could leak into the abdominal pelvic cavity, for instance, and start eating holes through your other organs. And sometimes this actually happens in the example of what we call the peptic ulcer. Okay, and we'll actually talk about that when we get to the chapter on the digestive system. Absorption is simply what? Taking in stuff from the outside and internalizing it inside the cell, or allowing it to cross a layer of cells to get from one place to another. An example of the absorptive property of epithelial tissue is seen in the lungs. The lungs are made up of special air sacs called alveoli, and those alveoli are bordered by simple squamous epithelium, which is one layer of thin cells, extremely flat, that are very good at allowing gas to move from the air into the blood, and also allowing gas to move from the blood into the air. The gas that moves primarily into the blood in the respiratory system is oxygen. The gas that moves into the exhaled air from the blood is called carbon dioxide. And this is vitally important to keeping the body going. Without oxygen, we wouldn't be able to make enough ATP to have life continue. And if we didn't get rid of the carbon dioxide that we create as a byproduct of metabolism, as a waste gas, then we would cause the body's pH and other processes to shut down, and we'd also die. Okay, so that's an example of absorption. Other places where you see absorption are places like the gastrointestinal system, say in the large and small intestine. All right, and there's other examples as well. Filtration. We talked about filtration when we talked about cell transport. Filtration is simply the ability to allow certain molecules to pass through a barrier while other molecules cannot. An example of filtration, a really good example, is seen in the ventricles of the brain, hollow chambers inside the brain that are filled with a fluid called cerebrospinal fluid, which is actually a filtrate of the blood plasma. What happens in these special locations inside the ventricles of the brain, called the choroid plexus, is that the pressure of the blood against these filtration membranes causes water and molecules below a certain size to enter the nervous system, the central nervous system, and create an environment that keeps the nerve tissue happy and healthy without allowing the larger components of blood to pass through that same barrier. Things like the red blood cells and white blood cells and platelets, and also potential pathogens, aren't allowed to pass through this choroid plexus because of the size of the openings that would allow these molecules to get through. Okay? Another example where you see filtration is in the kidney, which is tasked with cleaning your blood. As a result of normal life processes, the blood fills up with waste products, things like carbon dioxide, urea, and ammonia accumulate in the blood. And it's the kidney's job to primarily get rid of what we call nitrogenous waste, things like ammonia and urea. If they were allowed to build up inside the blood and inside your body tissues, we would upset the pH of those body tissues, and we would shut down enzymatic reactions, and we would also cause membrane potential to get screwed up. Okay, so, and again, another very important area in the body where filtration takes place. Secretion is the production of a cellular product that is then either released onto an open surface or inside the chamber of an organ. Okay, areas where you see secretions include places like the upper respiratory tract, which is lined with a very special type of epithelial tissue that's called, and get a load of this, pseudostratified ciliated columnar epithelium. And I know that that's a mouthful, but if you break the word down into its component parts, you'll get meaning out of it. Pseudo means fake, stratified means more than one layer, and ciliated means has projections, finger-like projections, on one surface of the cell, in this case what we call the apical surface. So pseudostratified ciliated columnar epithelium is tissue that looks under the microscope like it's more than one layer, but it's really only one layer, and it has cilia at its surface. And this particular type of tissue is responsible for secreting mucus, which is a sticky extracellular secretion that helps to trap particulate matter in the air that we breathe so that it doesn't end up in the lungs where it could cause irritation, inflammation, and scarring. Other epithelial tissues that are involved in secretion include the endocrine glands. Okay, they are tasked with secreting chemicals called hormones that help send signals throughout the body so that it can maintain homeostasis, keep itself in what we call that sense of balance that keeps us healthy, maintaining things like our fluid levels and our body temperature, our fuel levels, and our electrolyte levels, just to name a few important measurable characteristics that have to be maintained within a nice tight window in order for us to stay alive. Okay, so that's sort of a charcoal sketch of what some of these functions are for. If you were to say, okay, well, if I were to see epithelial tissue, how would I know it from any other type of tissue? A whole lot of cells very closely sealed together, okay? So if you look at epithelial tissue, it has lots of cells in it and very little extracellular space. So they tend to butt up against each other. These cells fit closely together. They often form sheets. The apical surface is what we call the free surface of the tissue, and the lower surface of the epithelium rests on what we call a basement membrane, which is actually the product of another type of tissue that we'll touch on in a bit called connective tissue, which is the body's glue. Many of these epithelial tissues tend to be avascular, meaning that they don't have a blood supply nearby, and as a result, they either have to get their nutrients through some other means, or it's intended that those particular epithelial tissues will eventually die and be regenerated, which is the case in most linings of organs. And for instance, your skin is constantly being shed and renewed on a daily basis. It regenerates easily if it is well nourished. And what you're looking at here are some examples of epithelial tissue classed according to their layers. Okay, so you can see here what simple epithelial tissue looks like. That has one layer of cells next to each other, and underneath them a layer of connective tissue. In this case, you might find this type of tissue, for instance, in the lungs, okay, as an example. This is what we call simple squamous epithelium. These cells are flat like a pancake, and they lie one right next to the other. Here's an example of what we call stratified epithelium, more than one cell layer, and the cell layer is named according to the shape of the cell in the most superficial layer. Okay, so this would be stratified squamous epithelium because the top layer tends to be pretty flat. Okay, this would be the basal surface of this tissue, and this would be what we call the apical surface, or the free part. Okay. Here are some other ways that we can categorize cells. We can categorize them according to shape. Squamous cells tend to be flattened like a pancake, and cuboidal cells tend to be shaped like a cube. They tend to be as tall as they are wide as they are thick, while columnar cells tend to be taller than they are wide or thick. And they all have a nucleus. You can see the nucleus here in the center of each of these cells. And they're all found in slightly different areas of the body. And we'll see a couple of examples here as we tour these different tissue types. So this is an example of simple squamous epithelia. It's a single layer of flattened cells. Locations usually are found in areas of the body that we call membranes. Now this is a different use of the word membrane than the cell membrane. Here a membrane means a sheet of flattened cells. Examples of membranes include things like the peritoneal membrane, which lines the abdominal pelvic cavity and secretes a serous fluid and has a lot of blood vessels in it. Most of these types of tissues line body cavities. They line the lungs and the capillaries, and they function in diffusion, filtration, or secretion in different types of membranes. Okay, so for instance, if we were to look at the cross section of a blood vessel, okay, we would find that the inner lining of the blood vessel would have a layer of cells that looks a lot like this, and that layer of cells, it has the ability to repel the formed elements of blood and at the same time remain functionally intact. Okay, so the blood's going to stay inside the vessel as it flows past. Other places that we find this type of tissue, this particular picture is taken from the alveoli of the lungs. Okay, and so you can see here these air-filled chambers that are surrounded by simple squamous epithelium, and this is designed primarily for gas exchange. So as we move through each of these different tissue types, I want you to think about, number one, what the cell looks like, number two, where we find it, and number three, what its job is. And remember what we talked about in lecture earlier, where we said that in engineering, form follows function, meaning that the reason that a cell has a particular set of characteristics is because it has a particular job to do. Okay, in this case, the dimensions of the cell lend it to being very good at things like diffusion, filtration, secretion, and protecting layers of the body. Here's another example. This is simple cuboidal epithelia. This is a single layer of cube-like cells. We find this type of tissue in glands and lining their ducts. A duct is simply a hose that leads from a gland into another location. It forms, for instance, the walls of kidney tubules. It covers the ovaries, and it functions in secretion and absorption. Ciliated types propel mucus or reproductive cells, the eggs in the female and the sperm in the male. What you're looking at here in this picture is a cross-section through kidney tubules. Remember, I told you that the kidneys have as their main job the cleaning of blood, and this is simple cuboidal epithelium. These tubes are being cross-sectioned in the figure that you're looking at here. This is what we call the lumen of the tube. This is the space inside the hose, and these are the simple cuboidal cells that surround it and are involved in secretion and absorption. Okay, secretion spitting products out into the lumen in this case, and absorption collecting stuff from what's ever inside the lumen and then transporting it to another location. Okay, very good at those particular tasks. Here's an example of simple columnar epithelia, and this is taken from the gastrointestinal system. You can see that these cells are taller than they are wide or thick. They often include mucus-producing goblet cells scattered amongst the cells of the columnar epithelium, and the mucus-producing cells make mucus, right, which is a sticky fluid. It is what we call a glycoprotein, and its job is to lubricate in the case of the digestive system. In the case of the respiratory system, its job is to trap particulate matter. Locations where we find it: it lines the digestive tract. It also lines the upper respiratory system. It functions in secretion and absorption, and ciliated types again propel mucus or reproductive cells. Okay, this particular slide is taken from a section of small intestine. Okay, small intestine is important because it's an organ that's vital in extracting nutrients from the food that you eat and taking those simple building blocks of nutrients, in the case of proteins, the amino acids, in the case of carbohydrates, the simple sugars, and in the case of fat, the fatty acids and the glycerol, and transporting them across their surface and into the bloodstream, where they'll be carried first to the liver, which will decide either to store them for later use or to pass them on to the rest of the body to use as fuel if there's an immediate need. Okay, so again, an example of where these types of tissues are found. Okay, here's another example. This is pseudostratified ciliated columnar epithelium, and you can see that it looks like it's more than one layer of cells, but it in fact is only one layer of cells. And the reason for that is that some of the cells are very short and some of the cells are very tall and lean over the short cells. They have cilia at their apical edge, and the cilia are important in propelling mucus. This is taken again from upper respiratory tissue. It looks like a double layer of cells, but all the cells rest on the same basement membrane. We find it again in the reproductive tract. We also find it in the upper respiratory, and where it functions there again in absorption and secretion. Okay, primarily in the upper respiratory, it's involved in secretion. Okay, stratified squamous epithelia are layers of flattened cells, one on top of the other, and they have primarily a protective purpose. The cells at the apical end are flattened. The apical end, remember, is the free area. Okay, there's nothing but empty space up here, and then this is you from here on down. It functions as a protective covering where friction is common. And where do we find these types of tissue? We find them in the lining of the mouth, the skin, the esophagus, just as a few examples. The mouth and the skin you're familiar with. The esophagus is the tube that connects the oral cavity to the stomach. So this is how food gets from your mouth into your stomach, and from there it goes to the rest of the digestive system. And so you can see here this layer of tissue These are the stratified squamous epithelial cells. And then down here, we have something interesting called a basement membrane. Okay, the basement membrane is connective tissue, and this layer of cells here is called the germinative layer or the stratum basale. And what it does is it constantly regenerates new skin cells as the old ones move towards the surface and die and are shed off. And the reason that they die is because they move further and further away from the blood supply in the connective tissue layer, and eventually they get so starved for nutrients and oxygen that they simply throw away all their cellular organelles and commit suicide. And you might think, well, this is a terrible thing, but actually it's not, because their job is to provide a protective layer between you and the outside world, which they do very effectively, because even though they're dead, they're held very tightly together, so they form a nice waterproof barrier and a barrier that's impenetrable to most pathogens. In addition to that, the fact that they're constantly shed keeps infections from setting up. If we didn't shed our skin on a daily basis, we would be much more prone to things like mold and bacterial and viral infections, as they would set up house on the skin and potentially erode away these outer layers and get down deeper into the connective tissue and eventually get into the blood, where they would make us really sick really quick. Okay, so again, form follows function here. There's a reason that these cells look and act the way that they do. Okay, protective layer. Stratified epithelia come in several forms. Stratified cuboidal epithelia are where you have two layers of cuboidal cells, and these primarily function as protection. Stratified columnar epithelia are where the surface cells are columnar and the cells underneath can vary in size and shape, and they also function in protection. Stratified cuboidal and columnar tissue are rare in the human body. We find them primarily in the ducts of extremely large glands. Now, stratified epithelia here we're looking at a unique example. We talked a little bit in lab about the fact that we would discuss transitional epithelia, which is what you're looking at here in lecture. And transitional epithelia is special because it has the ability to change its shape as it gets stretched out. It's made up of modified stratified squamous epithelium. The shape of the cells depends on the amount of stretching that that tissue layer is experiencing. And we find this tissue primarily in the ureters and in the bladder, which are parts of the urinary system. So they have the ability to change their shape depending on how stretched out they are. They function in stretching and the ability to return to their normal shape. We find them primarily lining the urinary system, and that's important because the urinary system experiences a lot of changes in volume as it performs its normal homeostatic function, which is to maintain fluid balance in the body and remove nitrogenous waste via the urine stream. And so you're looking at here a slide of transitional epithelium. You can see here the connective tissue underneath it, and then above it you've got these several layers of epithelial tissue. In this case, the tissue isn't distended, it's not stretched out. If the tissue were stretched out, it would appear squamous on the micrograph. Okay, a glandular epithelium is a tissue that secretes chemical signals or chemical products that the body needs to maintain homeostasis. A gland is one or more cells responsible for secreting a particular product. The secretions contain protein molecules in an aqueous fluid in many cases. Okay, and the reason for this is that proteins, due to the fact that they are what we call polar molecules most of the time, dissolve very well in water, and as a result they can carry out their signaling function in an aqueous environment. There's two major types of glandular epithelium. Endocrine glands are what we call ductless because they don't have a hose that allows their products to get to the outside world. Instead, what they have to do is secrete their products directly into the bloodstream using one of those forms of membrane transport that we talked about earlier in the lecture. Where an exocrine gland empties its secretion through a hose, a duct, directly onto a surface or into the lumen of another organ. Examples include the sweat glands and the oil glands in your skin. Okay, there's also glands that line the stomach, for instance, gastric pits that secrete hydrochloric acid and pepsin that help us digest the food that we eat. Another example of an exocrine gland. Okay, there are also organs in the body which have features of both. An example would be the pancreas. The pancreas contains both exocrine tissue, which secretes digestive enzymes that help us break down the food that we eat directly into the small intestine. And also, if you look at it under the microscope, it contains islands of glandular tissue. The major glandular secretions of the endocrine function of the pancreas include two hormones that control sugar balance in the blood. Those are insulin and glucagon. Okay, so you can have organs in the body that have features of both exocrine and endocrine glandular tissue. Connective tissue is the body's glue, mostly. Okay, it's found everywhere in the body, and it's the most abundant and widely distributed tissue. It binds body tissues together, it supports the body, and it provides protection as well, just as the epithelial tissue does. It has variations in blood supply. Some connective tissue is well vascularized, and some have a very poor blood supply or are said to be avascular, and we'll look at examples of those a little bit later on. The most important aspect of connective tissue is that it has a lot of extracellular matrix, which is a non-living material that surrounds the living cells, and they primarily provide the function for the connective tissue. Okay, it's the extracellular matrix that's where the action is as far as connective tissue is concerned. And in this sense, histologically, they're sort of the opposite of epithelial tissue. You have very few cells and a lot of extracellular stuff in connective tissue, whereas in epithelial tissue, you have a lot of cells packed very closely together and very little extracellular matrix. So that's how you can distinguish the two cell types. There's two main elements to extracellular matrix: ground substance, which is mostly water, along with adhesion proteins and polysaccharide molecules. Polysaccharides are sugar chains hooked one to the other. Fibers are produced by these cells, and there's three main types of fibers that we see in what we call connective tissue proper. Those include collagen fiber, elastic fiber, and reticular fiber. Okay? Collagen fiber is very tough stuff. Examples of tissues that contain a lot of collagen include hair and nails, just a few examples. Elastic fibers are found in connective tissue such as the cartilage on the outside of your ear, okay, and at the tip of your nose and in the flap that covers the opening to your trachea, known as the epiglottis. Reticular fibers are smaller, and what they do is they primarily maintain the shape of the connective tissue, and they also lend strength. So here's our first type of connective tissue. It's something you've probably seen before. This is bone. Bone is what we call osseous tissue, and it's made up of bone cells called osteocytes, which sit in these little chambers that are called lacunae, which are cavities inside which the bone cells get trapped after they secrete their extracellular matrix around them, and it traps them forever. And a hard matrix of calcium salt is what dominates a lot of the extracellular matrix around these osteocytes. There's lots of collagen fiber in bone as well, interdigitated with the calcified matrix, and it functions to protect and support the body, and it also allows us the ability to move through structures known as joints. And so you can see here an example of a special type of bone tissue called compact bone, which is the layer of hard tissue found at the surfaces of long bones, flat bones, and irregular bones throughout the body. This is a diagram of it, and this is an actual photomicrograph of compact bone. And what you can see here are the lacunae, which contain the osteocytes. You can see the lamellae, which are these rings of calcified tissue that have a similar center, so they're said to be concentric. This structure is the central canal through which a blood vessel will run that provides nourishment for this entire structure that I'm circling here, known as the osteon. And then each one of these concentric circles is called a lamella, okay? So that's a lamella, and that's a lamella, okay? And that's a lamella, and they're all concentric, okay? They all surround this same central canal. And so the way that this happens when bone is being formed is that initially all the bones in your body start out as cartilaginous material, which we'll see in a minute in the next slide. And then what will happen is that blood vessels will bring in bone-building cells that will at first tear down the cartilage and destroy it and replace it with this bony tissue. And as they do, they get trapped inside their own bony secretions. And at that point, the only way that they can get nourishment is through these tiny cytoplasmic extensions that protrude out from the osteocytes and reach across the lamellae so that they can communicate with each other and share stuff like nutrients and eliminate waste products, and also at least be reasonably close to a store of oxygen and nutrients provided by the blood vessels that not only run through the center of the osteon but also run across the osteon. If we had a different view of this tissue, a sagittal cut, we could see some of those what we call perforating canals. And then eventually all the cartilage in the bone is replaced by this either compact or spongy bone. The interior of the bone contains a special hollow area called the marrow cavity, which in long bones contains basically fat tissue, and in the bones of the pelvis and the rib cage and the sternum and the scapula contain what we call red bone marrow, which is where red blood cells are made. By the time the bone is mature, the only remaining cartilage is found at the tips of the bones, okay, where near in the case of the long bone, the tips are called the epiphyses, and those pieces of cartilage are called articular cartilages. And then also in a very special structure just behind the articular cartilage known as the epiphyseal plate, which persists until long bone growth stops by the time puberty is over. And after that event happens, after that little plate of cartilage is gone, no more long bone growth is possible, and at that point all that happens is that we begin to get shorter as our skeletal system begins to compress. Okay, this is hyaline cartilage. It's another type of connective tissue. It's found throughout the body. It's the most common type of cartilage, and it's made up of collagen fibers, and it also contains collagen cells that we call chondrocytes. The extracellular matrix is very rubbery. This is very flexible connective tissue. We find it in the larynx. It makes up the entire fetal skeleton prior to birth, and it functions as a more flexible skeletal element than bone does. And you can see a similar arrangement in one sense to bone in that the chondrocytes are also trapped inside their extracellular matrix. These are lacunae, just as the lacunae in compact bone trapped the osteocytes, the lacunae in hyaline cartilage trap the cardiocytes. And the only way they can get nutrition is through diffusion of oxygen and nutrients through the extracellular material into the actual chondrocytes. So this is why even as we age, we should remain somewhat active, because that movement of our joints and our skeletal system helps to promote the diffusion of synovial fluid, the liquid inside movable joints that allows them to move freely. Inside the joint cavity, that helps to perfuse the hyaline cartilage and provides nutrition for the chondrocytes and keeps them alive. As we age, our hyaline cartilage at the tips of bones tends to break down and eventually exposes naked bone inside the joint cavity, and in freely movable joints this results in an inflammation of the joint known as osteoarthritis, which is the arthritis that comes as a result of a lifetime of use. Another type of connective tissue is called elastic cartilage. Okay? It provides the ability to stretch, and it's found in the external ear, Fibrocartilage is highly compressible, and it's found in cushion-like discs between the vertebrae, okay, called intervertebral discs. So here you can see a picture of fibrocartilage. These are the chondrocytes locked inside their lacunae, and this is just an example of where the intervertebral discs are found. They're found between the vertebra. The vertebral column you probably know better as your backbone. And if you've ever heard of somebody having a slipped disc, what happens in these individuals is when they attempt to lift a heavy object, the force that's produced at one end of the intervertebral disc will cause the liquid center of the disc to bulge out through the annulus, which is made up of fibrocartilage, and that can press on the spinal cord, which is that piece of nerve tissue that carries signals from the brain to the peripheral nervous system, which carries electrical signals to the rest of the body. And if that happens, what can occur is that we can have numbness and tingling at areas that are distal to the area of the slipped disc, or in a worst case scenario, we can even have a damaged spinal cord that could result in some cases in paralysis. Okay, so just a little sidebar here on the intervertebral discs, and we'll talk more about those when we actually talk about the skeletal system. And here you can see a picture of the fibrocartilage that makes up an intervertebral discs. You can see the chondrocytes embedded inside their lacunae, and you can see the collagen fibers running throughout the matrix. Okay, this is dense regular connective tissue, and we find this primarily in ligaments and tendons. Ligaments bind bone to bone, and tendons bind muscle to bone. Dense regular connective tissue is a very fibrous type of tissue. The main matrix is collagen. The fibroblasts are the cells that make the fibers, and we find this again in ligaments and tendons, and we also find it in the dermis, in the lower layers of the skin. And again, it provides support and reinforces, in this case, the joints, and also helps to provide movement as it serves as a way for bones to attach to each other across joints, and also muscles to attach to bone across joints. One of the things that you'll notice about dense connective tissue, if you look at real dense connective tissue in a living specimen, is that it's very pale in color, which means it has a very poor blood supply. And as a result, if it's torn or damaged, it doesn't heal very easily, because what we have to remember is that the blood contains the cells that help repair injury. And so if you have a very poor blood supply, the ability to knit back tendons is quite limited. That's why when you sprain your ankle or you twist your knee and you injure a tendon, it can take a long time to repair. In the case of ligaments, the blood supply is even poorer. Ligaments often look jet white if you observe them in living tissue. And when you rupture a ligament, often the only recourse is to surgically repair the ligament or replace it. Because of its limited ability to fix itself, it's unlikely that the injury will resolve on its own. And here you can see a picture of dense fibrous connective tissue. You can see the collagen fibers running through it, and you can see the fibroblast nuclei between these fibers. This is loose connective tissue, and what we're looking at here is what we call connective tissue proper, or areolar connective tissue. It's the most widely distributed connective tissue in the body. It's a soft and pliable tissue. Under a microscope, it looks like a collection of cobwebs. It functions as a packing tissue, and it contains all the different fiber types: collagen, elastic, and also reticular fibers. It can soak up excess fluid, which can often lead to edema in certain cases. Where do we find this type of tissue? We find it underneath the epidermis of the skin. We find it in the dermal layer, where it serves to attach the overlying skin to the underlying muscle. And we also find it between muscle tissue, where it serves to reinforce and strengthen the muscle so that when it contracts and produces force, the muscle itself stays together without pulling itself apart. Okay, so very good supportive type of adhesive that the body uses to try and hold different organs together via sealing their tissues to each other. So here you can see the fibroblast nuclei and the collagen and elastic fibers. And then if you look real close, you can see some of the reticular fibers as well. They're quite tiny. Okay, adipose tissue is a special type of loose connective tissue. The matrix is an areolar tissue that has fat globules, which are basically triglycerides. The reason that triglycerides are so important in human physiology is because they're major energy storehouses. There's lots of chemical potential energy locked up inside triglycerides, which we know as fat. This is why the body chooses to use it as a storage molecule for energy rather than something like a sugar or protein, because fat, due to the fact that it's hydrophobic, it doesn't mix well with water, tends to take up less volume and hold more calories than any other molecule type. Many cells contain large lipid droplets. You can see here in the micrograph that the cell itself is simply this yellow component here. That will be the nucleus, and this is the fat droplet. So the cell is basically squeezed against its own membrane. The nucleus is squeezed against its own membrane here. What does it do? It insulates the body, it protects organs, and it serves as a site of fuel storage. It also helps us maintain our body heat, and it also provides padding and protection for a lot of our vital organs. For instance, if you were to look at the heart, you would see a lot of adipose tissue around the heart that serves as padding and protection. You find it under the skin. Here you're looking at a picture of it from breast tissue, okay, and you can see here again the nuclei of the fat cells and the fat droplets. It tends to be avascular, okay, not a lot of blood vessels running through it. Here is loose connective tissue. It's a reticular tissue, meaning it has a mesh-like arrangement of fibers running through it. This delicate network of interwoven fibers provides structure and support. The locations include the stroma of lymphoid organs, as well as lymph nodes, the spleen, and the bone marrow. And what you're looking at here is a picture of spleen, and what you can see spaced throughout here are the reticular fibers. These are white blood cells, these purple staining things, and then you can see the actual reticulocytes scattered throughout as well, okay? And they're what are actually secreting these fibers that hold the other tissues in place. Blood is one of two types of connective tissue. The other liquid connective tissue is lymph. Blood is a vascular tissue. Blood cells are surrounded by a fluid matrix called blood plasma. Fibers are visible only when the blood clots, and that's the reason for blood clotting, again, is to prevent hemorrhaging, massive loss of blood through an injury. It functions as a transport vehicle for a wide variety of materials, including nutrients, hormones, waste products, including things like urea and ammonia and carbon dioxide, as well as oxygen. It's the binding of oxygen to a very special pigment in the red blood cells, the erythrocytes, shown here, that gives blood its red color. Okay? And you'll notice something interesting. Number one is that, again, this is a connective tissue defined by the fact that there aren't that many cells and there's a lot of extracellular stuff all through the tissue. But notice that the white blood cells, the neutrophils, the eosinophils, the basophils, the lymphocytes, and the monocytes all have nuclei. They're all capable of cell division, and they have organelles, and their job primarily is to fight disease, while the red blood cells and the platelets are devoid of membranous organelles. They lose those structures because those organelles actually wouldn't serve their greater purpose, which in the case of the erythrocyte is to transport oxygen and carbon dioxide, and in the case of the platelet, to promote blood clotting. Okay, muscle tissue has its primary task to produce movement. And there's three types of muscle tissue: skeletal, cardiac, and smooth. Uh, they all produce movement. The movement is produced by very special muscle proteins called actin and myosin. And here you can see an example of skeletal muscle. Uh, Skeletal muscle is unique in that it is voluntary and striated. The voluntary means that we tell it what to do. We dictate when it contracts and when it relaxes. And the striations refer to these alternating light and dark bands that are seen throughout the tissue section. The reason for these alternating light and dark bands is due to the fact that the actin and myosin overlap in a contractile structure known as a sarcomere. And it's when these fibers overlap each other that you get these dark regions when you look at this tissue under a microscope. We'll talk more about actin and myosin when we actually get to the chapter on how muscle contracts. But if you want just a basic idea of how actin and myosin interact with each other, what they do is that they lie side by side. We can do a little drawing here. If I were to draw a sarcomere, we can sort of sketch this out. Imagine that these are the thin filaments, okay, and then we'll draw over here another block of thin filaments. The thin filaments are called actin, and then we've got some thick filaments here. So let's take that, and we want to let's see here. Okay, and so we'll draw these thick filaments. These thick filaments are the myosin. Okay, so we draw myosin here. Okay, and the myosin has little projections on it called heads, and what these heads do is when the actin is in just the right conformation, the myosin ratchets that I'm drawing here will reach out and grab the actin and pull on it using ATP as an energy source, and as they pull on the actin, they'll cause the entire muscle fiber, sorry about that, to shorten. Okay, so draw these here, here, and here, and here. Okay, and so what happens when these guys grab and pull is that the entire muscle contractile unit, the entire sarcomere, will shorten in this direction. Okay, they'll get pulled towards the center, and as a result, the entire muscle fiber gets tinier using ATP as an energy source, and that's how the muscle produces movement. Okay, so just an idea. And the reason you see these dark bands in the micrograph is because that's where the overlap of the actin and myosin occurs in these repeating units. This unit would go on and on and on and on until it gets all the way to either end of the muscle fiber. And we'll talk more about that when we actually get to the chapter on muscle contraction. Now, cardiac muscle is under involuntary control. We don't tell it what to do, and it's found only in the heart, and its function is to pump blood. It is found in cardiomyocytes, okay, heart muscle cells, and the interesting thing about the cardiomyocytes, if you look at them here, is that they are held together by special cell-cell junctions called intercalated discs, which are going to contain tight junctions which keep the heart muscle tissue held together as it beats, but also contain little tunnels called connexons, which are gap junctions which allow electrical